I guess we can start now.
Common group which we had. Yes, please start the session. Or
Yeah.
else lot of
Okay. So first of all, guys, really I would like to apologize for yesterday. It was a very unfortunate incident with regards to the network. But anyways, we will still try to cover everything. So don't worry about that. Okay. So for today, we will begin with our R session on DESeq2. After we are done with our R session on DESeq2, we will have a very small theory session on EdgeR. We will mainly learn how the EdgeR normalization works. That is the main agenda. After which we will also try to cover the EdgeR pipeline in R. I have shared all the required R scripts with you. It should be there in the drive under scripts. As well as that, I had also shared the data required last day only, right? So if you would have been able to run the previous R script that we used for the pre-processing, you would have gotten the counts matrix that we need today, which is for coding. If you haven't been able to run it, no worries. Please try to run it in your own time to create the coding matrix, to create the matrix with only the coding genes. Okay. If you have any worries or if you have any errors with regards to that, reach out to me on Telegram. I will help you out. But I would encourage you to do that. I will not be uploading that because I want you guys to go through that pre-processing step. Okay. Um, I hope all of you have downloaded the script as well as the required data. So for today's class, I will share my screen and show you guys. So for today's class with respect to DSeq2, the first thing that we will need is the DSeq2 metadata.csv. Okay, we'll need this file, and as well as that, we will need the script. So the script is DGEA DSeq2.r. While you're at it, please also download the DGEA edgeR.r. But for this, we don't need an explicit metadata file. Okay, so give me a thumbs up if you have downloaded three things: the two R scripts, which are the DGEA DESeq2.r and DGEA edgeR.r, as well as the data for metadata, which is the HC versus LAA metadata.csv. These three things we will need for today's class. If you haven't downloaded them, go ahead and download them and give me a thumbs up. I am repeating again, if you were not able to run the previous pre-processing counts matrix.r script properly, you will not have the coding counts matrix, which is okay, because I want you guys to run that yourself, figure it out. Okay, if you face errors, obviously put it on Telegram, I will help you out. But I want you guys to get to the coding counts matrix yourself. I will not upload that. Okay, I saw quite a few thumbs up, so I guess we can begin with the analysis. Okay, so everybody go ahead and in R studio, open the DGEA DSeq2.r and make sure the two files, your counts matrix coding.csv as well as your HC versus LA metadata.csv, they are in the folder that you are putting inside setwd. If you have the same folder structure as me, you don't have to worry. Obviously, you won't have the same folder structure as me with respect to these, right? These will be different for you, these will be more personalized for you. So make sure you make the correct path changes. Okay, because if you don't, then you will get some path related errors. I'll give you guys a minute to do that, to open all of this and do that, and then we will begin.
So Shobhadeep,
Yes.
first we see that DGCA, DGSEC word, right?
Yes, we'll first do the DESeq2 one.
Okay.
We'll do, yeah.
Uh,
Okay, let us begin, guys. If you can, split your screen and keep a look at both your code and my code. I will run. and you guys can run it along with me. If you don't want to burden yourself with that, you can just stick around on Zoom and watch me run it the way I am running it, and you can always refer back to the recordings and run it yourself the next day. Okay, whichever is suitable for you. I will run each line and explain what each line does. So we all know what setwd does. It basically sets the it basically sets everything up, right? The directory that we'll be working with. Don't really pay attention to these. I don't know why these came. These are not supposed to come here. These are supposed to come much later. But anyway, let us not worry about that. Then go ahead and call all of these libraries. So these are all the packages that we will be using for today's lecture. I had already given you the installation instructions for them. Don't worry if you haven't downloaded all of them, just follow along with the lecture. You can download and install them later and run through the scripts. You will have the scripts with you. Okay. Once you have loaded them, so this is not the first time I'm loading them in this environment. That is why it did not take much time. But if you are loading it in for the first time, you will see quite a few texts here in red. They are all just messages. Don't worry, it mostly is not an error. Okay, unless and until it pops up saying that there was an error loading this package, it is not an error. Okay, if you are facing an error, just follow the Zoom call for now, and we can talk about the error later on Telegram. Okay, now the first step is we have to load our raw final counts matrix, right? So we will be loading it onto this variable, and when we use read.csv, it will be basically converted to a data frame. Okay, now if I run this count command, I should now have this counts matrix here loaded properly. And the reason I used row.names equal to 1 is because in my CSV file, the first row was actually these gene names. If you remember, we saved it like that. We did row.names equal to true when we did the write.csv in the pre-process counts matrix. That is why the first row also got saved as part of the CSV file. And since we want the first row as the row names, we put row.names equal to 1 here. Okay. Once I have done that, um, now I will be starting with the analysis. So here we can see that we have three conditions, right? HC, LAA, and SAA. So HC stands for healthy controls. Your LAA stands for large artery atherosclerosis, which is a type of ischemic stroke. Okay. And your SAA stands for small artery atherosclerosis, which is a type of ischemic stroke as well. So the paper that I have taken this data from, they had compared amongst these three. For the sake of the example, we will now compare only between healthy controls and large artery atherosclerosis. Okay. Now you can see here that I'm using a very particular syntax here. So this syntax is specific to this library called dplyr. Okay, this is pronounced as dplyr, and this basically makes your data wrangling operations much easier. Okay, your data wrangling operations means like you're subsetting, you're mutating some columns, etc. So usually in base R we would have done that using subset, or we would have done that using data frame name dollar, then the column name and all of that. Dplyr just makes the syntax a bit easier in my opinion, and it makes it like apparently it works a bit faster as well than based on large data. So yeah, that is why I have used dplyr here. Don't worry if you don't want to use dplyr, you can also do these in base R. That is not a problem. But this is like everybody uses dplyr. That is why I wanted to give you guys a flavor of that as well. Okay, so here what we'll basically be doing is we'll basically be selecting the first 6 columns, because the first 3 are your healthy controls, and this fourth, fifth, and sixth are your large artery atherosclerosis. So if I run this line, I will now get my HC versus LAA matrix, which is my healthy control versus large artery atherosclerosis, and the row names are my genes. Okay. Now I also want to read in the metadata file. So the metadata file that I've shared with you, it has to be in this specific way only. If I read that in, you can see here that this is how it is made right now. So in the metadata file, actually, if you would have opened it using Excel, you would have seen here there is a sample column, and then these are the sample names. So make sure these sample names are the exact names that will be there in your columns of your counts matrix. Okay, that is very important for DESeq2. and then you will basically have another column here which basically tells the condition. And make a note of how you're writing the condition for each of them, because these will matter when we are setting up the dseq2 object. Okay. So once you have loaded both of these, and yes, this particular format is exactly what is needed for dseq2. dseq2 does not accept any other formats. Okay. So once you have loaded all of these, we will now create the dseq2 data object, right? So I have just we use for that, we use this function dseq dataset from matrix. Okay. And in the count data, we will give it HC versus LAA, which is our counts matrix. In the column data, we will give it the HC versus LAA metadata, which is our metadata matrix. And the design, we will give it as tilde condition. This tilde is very important. This is part of dseq2's syntax. Okay. So here basically, if you're only giving condition, that means this is the this is the condition column, right? So make sure this condition, how it is written with C capital and the rest small, it is also given here. Okay. So this basically means that this is your covariate of interest. Like this, you are going to be calculating all the differential genes based on these conditions. That is why you have given this. Okay. Now, just for the sake of this example, I have kept it only as a single covariate, right? Here, for example, in the metadata, if you had another column of, say, maybe gender, and there would have been males and females here, right? In the dseq2 formula, you are also allowed to use plus gender, so that the dseq2 model takes that covariate into account as well. And you can have differential genes expressed based on both this condition as well as based on your gender differences as well. So that is also allowed. You guys can check out the dseq2 documentation or the paper for that, how they actually do it. But since we are going to be learning the most basic RNA-seq pipeline here, I have not gone into all those complexities. Okay. Now if I run this, it should basically create the dseq2 object. And yes, you will see this warning message. You see this warning message because in the metadata you have mentioned control and LA as texts. So what dseq2 basically does is dseq2 expects numbers here. So ideally you would have 000 and 111 here, right? So if you don't have that, and if you have texts and the texts are matching for the 3 and 3, like here, dseq2 basically converts them into the numbers. See, it is saying that it's converting to factors, and it basically warns you that, okay, you did not provide me what I wanted, so I converted it myself. Okay. Now the next step is actually a filtering step that we do. So this filtering step ensures that genes across this row, genes which have a total count of less than 10, they are actually not considered in our analysis. Okay. So in this analysis, genes, for example, A1CF has a total count of only 2 across all of these 6 samples. So it is considered that A1CF is a very A1CF does not really provide too much of information across the 6 samples here, right? So if it does not provide too much information, we do not need to consider it for our analysis further. So this is a filtering step that we do. It is the recommended filtering step for any kind of differential gene expression analysis. Like you guys can also have your own definitions of filtering, right? So for example, some people might have that, okay, if a gene is less than 5 in any of the samples, you filter that out. Okay. Then some people might have it like, okay, if a particular gene is less than 10 in at least half of the samples, then you filter it out. So all of that is completely upon you. The lab that I worked in, this was the standard that we used to follow, and I have seen a lot of papers do this filtering as well. So this is kind of like a very acceptable way to filter out genes which are not as informative, which won't really give you a lot to think about. Okay. So again, reiterating, what this is doing is removing all genes that have a sum of 10 across all 6 samples, less than a sum of 10 across all 6 samples. So genes such as A1CF, which have a sum of only 2 across all 6 samples, will be removed. Okay. So once
Sir,
I
can
do
you
that,
also, sir, can you also repeat from line number 16 to 18, sir?
yeah. So
The
line number 16 to 18 is basically you defining your DSeq2 dataset object. Okay, so whenever you're working with DSeq2, you have to create this dataset object. Okay, and the function to do it is DSeq dataset from matrix. So this is something that is like central to the DSeq2 library. This There is not much to understand here apart from the fact that this is how the code is written in the DSeq2 package, right? Somebody has written a lot of code inside this package and they expect you to follow the rules that they have set. That is all there is to it, right? So the way to use this function is as follows. In count data, you have to give your counts matrix, and you have to make sure your counts matrix is actually only numbers. If you see, these are actually only numbers. The call names and the row names don't actually form a part of the matrix per se. Okay, so that is something you need to make sure. Your call data should be your HC versus LA metadata. Again, if you see, the metadata has only one column, which is condition, and only the conditions are part of my data or a part of my matrix. The condition column is actually, this is just the column name, and these are the row names, so they are not part of the matrix per se. Only the unshaded area is part of the matrix. And yes, these are rules you have to abide by, or else DSeq2 will throw you errors. Okay, and the last one is the design. So this is your experimental design, right? So here, since we are only considering the condition as the only point of difference here, right? So whether it is a control sample or it is a disease sample in our scenario, right? That is why we have put only condition here after this sign, which is known as the tilde. It will be on the top left of your keyboard, usually under the escape key. Okay. If, say, for some example, there would have also been a column of gender, right? And you would have male, female, female, male, whatever, right? Now you can also decide to add in the factor gender within your experimental design. Say, for example, you want to find out differences both with respect to condition as well as with respect to gender. Then all you would have to do here is write plus gender, right? And so these are basically complex experimental designs, and DSeq2 has a way for you to use complex experimental designs. If you just search on Google with DSeq2 complex experimental designs, you will see a lot of examples, but since they're a bit more complicated than the normal one, I have not used it here. Okay, so moving ahead, now that we have filtered, we can check
And
how many.
Shodadev, one more
Yes,
question. If I add, because we have to write condition, then plus, no
yes,
space, then suppose gender.
yeah, I mean, space and all, that is fine. If you even put
Okay,
a space here, that is fine, that won't matter.
okay.
Yeah.
And if that thing we add two conditions like
Yes.
this, should that affect the programming, the downstream programming? Do we have to
Down
change thing?
Downstream programming will be affected at the step where you actually uh, calculate this. So this is where it will be affected. Okay. Here,
Okay,
for example, since I have put only one condition, I am only using the difference based on condition. This is the other line that you will have to change, the results function. You
okay,
can
okay,
find it all in the vignettes. Okay,
okay,
just
okay,
take a look at it. Okay, so for the sake of this example, I've tried to keep it as simple as possible.
okay.
Okay, yeah. Okay, so now that we have filtered out the genes which, according to us, are not as informative, we will now see how many genes we are left with. So to begin with, we had 19,270 genes. After that, now you can see if I do an n row, that is the number of rows in DDS, I am only left with 13,357 genes. So around 6,000 genes, we have decreed them as not informative enough for our analysis. And this is completely acceptable. Even if you are left with anything above 10,000 genes after doing your filtering, you are okay. That is, I mean, 10,000 is like the ballpark number. Anything less than 10,000, maybe you are over filtering. Okay. Now we will do something called as a variance stabilizing transform. Okay, so here, as you guys have seen, we have not yet done any of the normalization steps that DSeq2 does. So actually in DSeq2, normalization happens as part of the differential gene expression analysis, so you don't have to do it manually, but we want to visualize the data first before we jump into differential gene expression analysis. Okay, so before visualizing, we need to perform this variance stabilizing transform. Why? Because RNA-seq data is very heteroscedastic. By heteroscedastic, I mean that the variance does not increase or decrease properly with mean. So the variance does not remain constant, right? If you remember, we had seen a plot where variance increases with increased mean expression, right? You guys remember there was a plot with all the black dots as genes and then a red line going through it. Go through the
Yes.
previous lecture, you will see that. So this basically tells you that RNA-seq data variance is not stable across mean expressions, right? And that will affect your PCA, because what your PCA does is your PCA tries to figure out the variance explained by each component, right? Your variance explained by each component as in each gene, okay? And then it tries to give you the overall distances between samples. Now, if I had some genes which were extremely highly variant and some genes which were extremely lowly variant, only the genes which are very highly variant would be driving my PCA, and they would be the ones who will be contributing the most towards my clustering in PCA. I don't want that. I want a method to stabilize the variance across mean expressions. I want the variance to not vary too much across high and low mean expressions. That is why I use something called variance stabilizing transform. So this has very complicated math behind it. We won't go into that, but always try to use blind equal to true. I mean, blind equal to true, I think it is the default only, but still I wanted you guys to show, because then it does not do any prior normalization techniques. It does it on the raw counts, okay? And that is the truest variance stabilizing transform that you should be doing, okay? Now if I run this, it will take a couple of seconds. Once it has run, we will be ready to do the PCA. Now DSeq2 actually has a function for you to plot PCA, right? It already has this function, and in this function you have to give the variance stabilized transform matrix, right? As well as the group by which you want the PCA to come back. So for example, in our case, we want the PCA to separate my samples based on condition, okay? And I have done return data true so that we get the data here and we can manipulate it here while plotting, okay? Now if I run this, you will see again this using n top equal to 500 top features by variance. That just means that it is only going to calculate the principal components for the first 500 principal components, okay? Don't worry if you don't understand too much about PCA. We will be going through PCA in more detail during our ML lectures, right? Even if you don't understand, go back to lecture number 2 where I gave you a brief description of what a PCA is. You should at least be able to grasp the idea behind it, okay? Now we have to extract the percentage variance explained, because we want to show it on the plot as well. So for that, I have written this line. So we can directly do it from the PCA data actually, right? And since so basically the percentage variance explained is in terms of between 0 to 1, so it will be in 0.12 or 0.13, like that. That is why we are doing a 100 multiplied, right? And this percentage variance actually, I think you can see here in your PCA data as well, right? No, sorry, this is basically your thing that you're giving it the name for, okay? So now if I see the percentage variance here, I have basically a variable which has these two numbers. These are your percentage variances explained, okay? Again, percentage variance explained just basically means that if I'll actually plot it first and then I will show you. So for plotting here, I am using something called ggplot. I understand that the syntax here is very difficult to understand. Don't worry, even I don't remember this syntax. I have to always go back to the documentation or Google or Stack Overflow to get the syntax and do it. But I will explain you what it means. Here, the first argument that you give it is actually your PCA data frame, okay, which you created using DSeq2. The second argument is AES. I don't even remember what it stands for, but here you have to give it what the X column will be, what the Y column will be. Basically, this is what is going to be printed, okay? This is here you define what your x column and y column will be from this PCA data. If you see here in the PCA data, we have PC1 and PC2. So this is what we are defining here. Your coloring of the samples will be based on your condition. Your condition is also part of this PCA data, right? And your labeling of the samples will be based on row names of PCA data. So if you can see here, the row names are actually our sample names. Okay. Then in ggplot, you have to give plus when you're actually adding stuff. So here geom point basically means, so for each sample, what is the size of the point, right? I have just put size 3. You can put anything as long as it helps you out. Okay. So this basically geom text repel is a function that is required for you to add the sample names on the plot. Okay. Then we are basically defining what will be our x axis label and our y axis label, right? So our x axis label will be, if you remember, pace 0 pace stuff, right? With respect to with just a space. So it will paste PC1, then the percentage variance that we calculated, and then the string percent variance. That's just it. This is labeling the x and the y axis, and gg title will give you the title of the plot, right? And I've put theme minimal here so that it does not use up a lot of the advanced graphic libraries that a lot of you might not have. Usually every system has the minimal graphics libraries which are required. Some Linux systems might not even have these, and you might face issues with these. In that case, you just have to go ahead and install those graphic libraries, because this is necessary for ggplot. Okay. Or else you can also plot PCAs without ggplot. You can do it in base R, but that becomes way too difficult. So yeah. Now if I run this, I should ideally see my PCA plot. So here I can see my PCA plot. As I said, the variance is printed here on the x and y axis as well. So this just basically means 43% variance means that along this axis, right, if I were to separate the samples along my x-axis, then that would be 43% fruitful. In the sense, it's not exactly what that means, but you can at least get an idea of what it means. It means that on 43% of your samples, which actually does not make sense, 43% of 6 is not a whole number, but you can roughly say that 43% of them will be actually separated out from each other. Okay. Similarly here for the y-axis as well. Now what I want you guys to see here is that you can see LA1 and LA3 are pretty close to each other, but LA2 is way far ahead here. Similarly, HC1 and HC3 are pretty close to each other, and HC2 is way out here, right? So these are probably your outliers. Okay, your HC2 is an outlier for the healthy condition, and your LA2 is an outlier for the LAA condition, right? Now, in the most ideal scenario, you should be removing them from any further analysis. You should not use them. And even if you remove them, you can see that there is a clear line I can draw here along the x-axis, right? That will separate these two particular clusters. Okay. But again, and this is why I chose this example, we are at a limit here, and this is why this is how I want to showcase to you guys the limitations we have. Even though we have such robust methods, we still have limitations. So the limitation here is that we only have 3 samples per group. If I remove a sample from each, I am violating the minimum criteria required for any robust statistical analysis. Okay. This is a bottleneck. In this scenario, even though I can see clearly I have outliers, I cannot really remove them, and I have to go ahead with the analysis. And this is a very real problem that a lot of labs face, because they only sequence the bare minimum. Okay, if I am going to study 2 conditions, a lot of labs will only sequence 3 for each condition, 3 samples for each condition, which is absolutely fine, because you also have to look on the economic side. Okay, so bear in mind, if we had 4 of each and we would have seen these outliers, we could have removed them. But since we only have 3 of each, we cannot remove them, although we should. Okay.
So, Shorodeep, by
Yes.
removing that thing,
Hmm.
what do you mean that we have to go back to the
Yes,
past and we do
you
the
will
whole
remove it from here.
selection, everything without that sample?
Yes, yes. So you have to, for example, you have to do the subsetting here so that you are only left with HC1, HC3 and LA1, LA3. You have to change the metadata file as well, because now you don't have 6 samples, you have only 4, and the rest of the stuff as well. So VST and PCA should also be performed again on that.
Okay.
Okay.
So from
Yes.
this RNA script,
Hmm.
we will delete that data, not from the beginning.
No, no, no. So you won't delete it from your file. Your file still remains the same. In the script, you are doing your deletion. Yes.
Yeah, yeah, yeah,
Yeah.
yeah.
Yeah. Yeah. Okay. So now that we have seen the PCA and we have understood how to actually plot it, we have gotten a rough idea what it means. Let us go now to the actual differential gene expression analysis. Before that, it is always advisable that you run this line first, which will basically tell you that these are your levels, right? So you have a control level and an LA level. So this is just a fancy way of saying that this is the distinction on the basis of which DESeq2 will now do perform the differential gene expression analysis. Okay. Now this particular function DESeq is what performs all the steps that we went through in the theory class last time. Okay. If I run this, you will see that it prints them as well, right? See, it is first, it estimates the size factors, which is the normalization step. It then estimated the dispersions. Then the gene-wise dispersion estimates were done. Mean dispersion relationship was plotted. Final dispersion estimates were calculated. And then finally, it fitted the generalized linear model and performed the hypothesis testing. Okay. This is the function that does that. And it takes in as parameter the DDS object that you had created here. Okay. This is the DDS object. Correct. Okay. Now that I have done this, this DDS object will now contain all the information that I need here. Okay. And here actually the main information that I need out of this, if I want to see, ideally now now want the table. I want the table with the log FC and the p values. For that, I have to run the function results names. Okay. If I run the function results names, oh no, sorry, my bad. So results names basically, so okay, this has not actually performed the hypothesis testing yet. Okay, we will be performing the hypothesis testing here in this step, my bad, sorry. When we actually perform results names, it basically tells you the condition that it is going to be testing for. So now we can see that the condition that it is going to be testing for is LAA versus control. Whichever is coming here on the right hand side of versus, this is what is DESeq2 going to be treating as the baseline. Okay. Ideally in your scenario, you should also have the control as the baseline. Okay, because this is your baseline expression. Now this is for a further step. I am actually going to extract the normalized counts and keep them for later use. We will be using these later. This is not part of the differential gene expression analysis pipeline per se, but this will be used further when we are doing some very important visualizations. So you just get the normalized counts out using the function counts, and then the normalized counts df you create it with the function as.data.frame on this. So normalized counts will basically be a matrix. So you're just converting it to a data frame in this step. If I run these, I should now have the normalized counts df. You can see here that I have the data frame which has the normalized counts. Okay. Now, in order for us to get the table, we are going to perform the hypothesis testing. So as I mentioned, by default, DESeq2 performs the Waltz test, right? We had gone through it. The way you define this contrast is very important. You always want the baseline to be first, then your condition, and you also want to you always want your sorry, you always want your column name to be first, because this defines the particular function, right? And in the beginning, so the second parameter per se should be your LAA, which is your condition, and the final parameter should be your control, which is going to be your baseline. So it should always be column name, condition, baseline, this
Sir,
method.
I have one question. Sir,
Yes.
here while normalizing the counts, so which
Hmm.
approach we are using? TPM?
No, so DESeq2 uses the median of ratios, right? We went through that
Okay.
in the last lecture. So this is what
Okay.
DESeq2 does by default. default, and that is what you should be using. Because if you
Okay.
remember, we only do TPM in case if we are going to be using t-test. Okay, since DC2 uses generalized linear models, it has its own normalization method that is best fit for that. Okay.
Okay.
Yeah. So actually the normalization happened when you ran this. Okay, at this step you are basically just extracting the normalized values. Okay. uh, so now if I run this results function with the definition like this, again remember, column, condition, baseline, this has to be your definition. If I run the results function, it will perform the hypothesis testing. Again, it took a couple of seconds, but now I have the results object. Now within the results object, okay, I will now get the first, what I will do is I will order it based on p-value. You can skip this part if you want, it does not really matter, but ideally you should, because if you want like clean looking results, that's it. Okay, so here what I'm doing is I'm ordering it based on p-value, and then I am also removing any na values. Okay, so once I have done that, I will now convert it to a data frame so that I can view it in my viewer here. So in order to convert it to a data frame, again, you have to run as.data.frame on the res object. If I do that, I should have the res df here. Yes. And so you see that it has calculated it for all the 13278 genes that we had. Okay. And so you can see that some of them have still been removed. Those were actually the na values, right? For some of them it probably could not calculate the calculate the log fcs or calculate the p values, or maybe the p values or log fcs are not actually defined in r. Maybe they are nan values or they are inf values, right? So infinity. So this is basically a limitation of the programming language. But again, you can see we have barely lost maybe what, 70 genes, maybe 60 70 genes. That is still fine. You don't really care about the 60 70 genes when you think about where you started from. You started from 20,000 genes, right? So that is still fine. Now I will put a p-value cutoff, right? Again, this resdf has these columns. So base mean is basically your baseline mean expression of that particular gene. Okay, your log2 fold change, as I mentioned earlier as well, this is basically how much it has changed, right? In So a log2 fold change of 1.69 means that in condition LAA there is a upregulation by log2 fold scale of 1.69 as compared to control. All the values here are expression values or are fold change values, where the control is the baseline and your condition is where it is being reflected. Okay, this is your standard error for the log2 fold change. So you, when you actually report it, you can report it as 1.69 plus minus 0.33, right? This is the wald statistic that it calculates, and this is the p-value with respect to this gene. This p adjusted is your Benjamin Hochberg false discovery rate corrected p-value. And when you are actually filtering, here you can see that I have put the filter as only p-value. This is actually wrong. I should ideally change it to p-adg, which means p adjusted. Okay, and I have put a p-value cutoff of 0.05. You can put whatever you want. Okay, now if I do this, I will now have a res p value cutoff, right? You can see that there are only actually 40 genes which pass the p adjusted cutoff, right? That is exactly why I had kept p value here, but no worries. This is what I wanted to show you guys what you should do here. Okay, I remember somebody asking the question that what if I get two less genes, right? If I use p adjusted, this is exactly what you should do. So the paper that I took this from, they have also mentioned that they have used p value instead of p adjusted so that they can make more sense of the biology. Okay, that is not a wrong thing to do. Okay, but it is going to be misleading if you don't report it. So if you happen to use p-value, please report it in your analysis. Okay, if you're writing a paper, if you're writing a thesis, wherever you're doing, report that you used p-value instead of p-adjusted, and make sure that you have a good enough explanation for it. Okay, having two less genes and not having been able to uh, g- come up with some biological inference with two less genes is a good enough explanation. So that is still allowed, but make sure you report it. Okay, so here I will keep p value only, but ideally if you have enough genes for p adjusted, you should use p adjusted. Okay, um, the you can ask how many genes are enough. There is no real correct or wrong answer to it. It is completely based on your analysis. For example, if your conditions, you thought that your conditions were very close to each other, right? Say for example, if in this scenario only I would have gotten only 40 differentially expressed genes in terms of LAA, which is large artery atherosclerosis, versus SAA, which is small artery atherosclerosis, that would still make some sense to me, right? Because they could ideally, from a biological standpoint, they are very close to each other in terms of conditions, right? But this right now we are doing LAA versus control, and I don't know enough biology about atherosclerotic strokes, right, to know whether they are actually pretty close to each other or not. But it would seem as so. But then again, since I don't know, and I want a biological inference out of my data, I am using p-value. Okay, by no means is this wrong or right. This is unethical to use p-value, but it is only unethical if you don't report it. Okay, please report it if you are using p-value. Otherwise, always use p-adjusted. So I will rerun this line so that it actually uses only the p-value. Okay, now you can see I have about 900 odd genes which are differentially expressed with respect to p-value, not p-adjusted. Okay, so I can also save this file because we will be using all of these later in our rest of when we are doing functional enrichment, when we are trying to figure out pathways which are over or underrepresented. So we can save this as well. Okay, I have a few extra lines here as well where you can put cutoffs of your own choice. So for example, I have put a log 2 fold change cutoff of 1.5. Here I have put a log 2 fold change cutoff of 1. Here I've put a log 2 fold change cutoff of 0.5. So you can make these and create these files and keep on your system. For this analysis, it's not really necessary, so I won't, but you can do that if you want. Okay, anyway, all of these will already be part of this file, so you can always just load this file and do the subsetting on that later. That is also fine. Okay, now anything that is less than 0.5 is worrisome. Okay, if your log 2 fold change is actually less than 0.5, ideally that means that it is not differentially expressed. Okay, this is something that researchers across the globe have agreed upon. Okay, again, it's not a hard and fast rule that you have to use a minimum cutoff of 0.5. You can define cutoffs based on your experiment. As I said, if your experimental conditions are very close to each other, you can also define a cutoff of 0.2 to be differentially expressed. That is up to you. But 99% cases, in 99% cases, people don't use a cutoff below 0.5. Okay, now you can see here that I have both a plus value and a minus value, right? So let me go here and actually show you guys what that indicates. So a plus value of log 2 fold change indicates that this gene is upregulated with a fold change of this in my condition, which is LA, as compared to control. Okay, now minus ones indicate that this gene is actually down regulated by this amount of fold change in my condition with respect to controls. That is why some log to fold chains will have a minus and some log to fold chains will have a plus. Okay, so this is really important to know and understand. Okay, so you can see here for a lot of them the p adjusted is actually na, and this is also one of the reasons why I chose p value, because if it's na, that just means that r could not approximate it, right? R could not calculate it properly, so that is not our
One thing I would like to ask here is that
yes, yes.
uh, um, when it is accurate to say that we need to take p value or p adjusted value. Suppose we have a
You
very small sample size, and on the other side we have a and data of from 100 individuals, in which case we will consider p value versus p adjusted value, small
You should all
population or large.
you should always consider p adjusted. Okay, only if you're not getting as many number of differentially expressed genes with a p adjusted cutoff, then only you should consider using p value. P adjusted
Okay.
is the one which is statistically correct. Okay, if you're not using p adjusted, you're technically not doing the correct thing. But then again, it is also important as a biologist for you to explain the biological phenomenon for which you need a good enough number of genes, right? Only 40 genes cannot explain a total biological phenomenon, right? That is the reason why I have taken p value here. You can also, but whenever you take, just report it that okay, instead of p adjusted I have actually used p value, and this is the reason why.
As we go up, we are going at a very stringent cutoff. For example, in that case, in your case, you have taken 0.05.
Yes.
So if we can take a 0.01, 0.05, this
Again,
doesn't mean, this doesn't have any significance if I am taking 0.01.
so
So I
0.01 would be even more stringent, right? You
yeah,
would have
definitely.
you would have even lesser genes with 0.01. So it is all based on how much error you are ready to accept. For example, if I am putting a p-value cutoff of 0.05, that just means that I am okay with having a 5% chance of calling this as a false positive. If I'm calling it as a differentially expressed gene, 5% of the times if I call it wrongly, I am okay with that. I am okay with that margin. Some people use a cutoff of even zero point 0.1. In that case, they are basically saying that if I call a gene as differentially expressed wrongly 10% of the times, I am okay with that. I will live with that. That is all there is to it. Okay. Again, these are also things you should consider before you start the experiment. In case of rare conditions, for example, when you have very less number of samples, it is very natural for you to have high p values even after so many statistical tests. So in case of rare conditions, people do use a p value of zero point 0.1, p value of as high as zero point 0.1. That is allowed. It, you should just know what it means. It just means that you are ready to take the risk of calling differentially expressed genes wrongly 5% of the time, or 10% of the time, or 15% of the time, or 1% of the time. Does that make sense?
So in many cases, I have seen that whenever we are taking uh, at the default value, whatever there is 0.05,
That.
we are not having any genes or zero genes in the Venn diagram.
Mhm. Mhm.
Uh, but what is the criteria to select the p value 0.01, 0.05, 0.1 particularly?
There is no defined criteria for that. It will be completely based on your experiment. As I said, you should be aware what setting a p-value means. If you are setting a p-value of 0.05, that means you are okay with making an error 5% of the times. So out of 100 genes that you called as differentially expressed, if 5 of them are actually not differentially expressed, but you flagged them as differentially expressed, you are okay with those 5 genes being wrong. Okay, that is you setting a margin. Okay, if you're okay with 10 genes out of 100 being called wrongly, that is you setting a margin, and that will be based on what your experimental design is. As I said, as an example, if it's a rare condition and if you have less samples automatically mean less statistical power. That automatically means you will have higher p values. So in case of experimental conditions where it is a rare condition, okay, you can ideally go up to a value of zero point 0.1 as your p value. Okay, I have seen papers who have done that. But if you are taking a p value that is above 0.05, because 0.05 is kind of like the globally accepted value, right? It is like the it is like the soft spot, it is like a sweet spot. Okay, if you are changing it from that, you should mention it, you should report it, and you should have a very good reason why. For example, the reason being less number of samples because it is a rare condition is a good enough reason for you to increase your p value cutoff from 0.05 to zero point 0.1. Okay, it is completely experiment centric, the choice of p value. The only thing you should keep in mind is you should decide on the p value before you even begin with the experiment. Before you begin
So
sequencing,
if
you
we
should
are
choose the p
definitely
value.
if we are cross comparing two transcriptome samples,
Mhm.
so is it mandatory to set the same p value for both samples, or we can select based on our choice?
No, no, no. Your p value, first of all, you get a were you there in last lecture when I explained how we calculate this p value?
I think yesterday there was no lecture.
No, not yesterday, day before yesterday.
I was there, yes.
Okay. Do you remember how we calculated the p value using Walt's test?
Yes, but
Yeah,
I need to just check it
yeah,
out.
that is fine. So I think there is a misconception here. You don't calculate a p value for each sample. Okay,
Yeah.
the p value is calculated for the gene's expression across all the samples. Okay, go through
Okay.
the lecture day before yesterday. Okay, I have explained how the p value is calculated. And so to answer your question, p value has to be same across all the genes. For every gene, your p value cutoff should be the same. You cannot have a p value cutoff of 0.01 for gene A and 0.05 for gene B.
No, no,
Then
no. I'm
you
not
are,
saying for one or two gene. We are
yes,
comparing cross comparing two transcriptome data set. And in
okay.
one transcriptome data set, I have
Hmm.
set the cutoff value
Oh,
0.05.
okay.
So
Yes,
we are
no,
getting the large number of genes. But in when I
you
am
should.
comparing that data set
Hmm.
with another transcriptome data set, the same
Okay,
samples of same
understood.
organism,
Okay, I
then
understood your question.
yeah, yeah.
Yes, in my opinion, as I have done it, I have done this similar thing where I've compared genes across two transcriptome data sets. You ideally, for statistical comparison, you should keep the p-value same in both. So if you're using
Okay.
a 0.05 cutoff for dataset A, you should also use 0.05 cutoff for dataset B. And whatever you get with it, you have to live with it. Okay. If you think that in dataset B, I am getting less number of genes with 0.05, I would get more with 0.1, then increase to zero point 0.1 for both. That is my advice to you. Even if you get too many genes in A, eventually when you try to make sense out of it using pathway enrichment or functional enrichment, they will boil down to only a few of the important genes. Okay.
Thank you.
Yeah. Okay.
Sir,
So
I have a doubt.
yes.
Sir, regarding the fold change, log to fold change,
Hmm.
whether we need to stick with log to fold change or fold change, sir?
uh, Sorry, I did not get your question.
Whether we need to report regarding the log to fold
Oh,
change
acha,
or fold change in,
reporting.
yes.
Some papers report log to fold change, some papers
Yeah.
report absolute
Yes.
fold change. You can just convert it as well. So log to fold change is basically what? Log to the base 2 of your absolute fold change. So if you have to convert it, you just have to do 2 to the power of this, right? Then you will get whatever is the absolute fold change. I think that is
Okay.
correct. Yeah.
Okay. So if I want to report, if I want to categorize based on the fold change means fold change greater than one or less than one means. So
Hmm.
accordingly, I need to scrutinize the genes, right, sir? Based
Absolutely.
upon that, I have to
Absolutely. If you
yes.
are using the log 2 fold change as your scrutinizing criteria, report that. Okay.
Okay.
If you're using fold change, that is absolute fold change as your scrutinizing criteria, you should ideally, after you get the log 2 fold change, after you get this data frame, res p val cut, convert it to
Okay.
absolute fold change, then do the scrutinization. Okay.
Okay.
So this
Okay.
filtering that you see, then do it on the absolute fold change. Don't do
Okay.
the filtering on log 2 fold change and then convert.
Oh, okay, fine. Because
Okay.
in some papers I have seen that if we set the category log to fold change as
Mhm.
0.58 5 means it means the
Mhm.
fold change is greater than 1. Is it right, sir?
Yes, if you think about it, that is the case, right? I think I will wait. Let me check once.
Okay. If we set as log to if the log to fold change is 1 means the fold change is 2.
Yes, yes, that is exactly
So
how it
based
is.
upon okay, so based upon that
Yeah,
we can set the log to fold change value.
yes, you can. I mean, the
Okay.
most commonly used ones are 0.1, sorry, 0.5, 1, and 1.5.
Okay,
Okay,
0.5, 1 and 1.5. Okay, so
1.5 are
based
the most
upon,
commonly used ones.
okay, okay. Can we use 0.585, 0.5?
0.5 and
Yeah, 0.5.
you can use 0.5. 0.5 is like the
Okay,
bare minimum. You should not go below 0.5. That is
okay,
like
okay,
the
okay, sir.
that is what people have agreed on. But then again, as I said, if it's a rare condition and you have so many sorry, if two conditions that you're actually comparing are very close, in that case
Okay,
you can even go below 0.5, right? Because then that would
okay,
be okay. Anyway, these conditions are so close, so I might not even see, so any subtle difference could actually be a biological difference.
okay, okay, sir. Okay, thank you.
Yeah. Okay. So once we are done with all of this filtering, again you should write them down to files. For that you use the function write.csv, but these are all that you can do on your time. This is not something that we need for the analysis, so I'll move ahead. Okay, now we want to inspect the top 25 upregulated and the top 25 downregulated genes. Okay, why would I want to do that? This is just to see that what are the genes which are basically giving the highest fold change, okay, given my statistical cutoff, so that I can say that okay, if these are the genes which are giving me the highest fold change, these are the genes which are the most different between the two samples. So maybe these are the driving forces of the differences, of the actual biological differences. Okay, so in order to do that, we can simply do a filtering again. So here I have defined a variable top 25 LA control. So again, this is a syntax which is used by dplyr. Okay, it allows you to basically pass the input of one part as an pass the output of one part as the input to the other. That is why, and in order to do that, you have to use this simple percentage arrow percentage. Okay, so this res p-value cut will be passed to this function filter. Filter will filter based on log to fold change value greater than or equal to 1.5. Now I'm putting here greater than or equal to 1.5 because I want the upregulated ones, right? That is why this is a plus 1.5, and p-value cutoff of 0.05. Again, this output will be passed on to the arrange function, and I want it to be arranged in descending order. That is highest fold change first, and I want only the top 25. That is why I'm using the head function. Okay, so if I run this, sorry, if I run this, I should get the top 25 upregulated genes here. These are my top 25 upregulated genes based on log fold change. Okay, where did it go? Yeah. Okay, as you can see, all of them here are uh, all of them here are upregulated because all of them have the plus sign. Okay, just give me a second, guys. I'll drink some water and come. Okay, let us continue. Similarly, I will also get the top 25 down-regulated genes, right? So these are the genes which were the most down-regulated in LAA with respect to our controls. It is the same logic to filter them out, but here only we are only considering genes which are less than or equal to minus 1.5. Okay, you could very well just keep here 0, right? Less than or equal to 0 or greater than or equal to 0. It will be the same behavior because all your filtering here is you're filtering for positive values, and here you're filtering for negative values. Okay, so the behavior will be the same even if you put 0 here instead of minus 1.5 and 0 here instead of plus 1.5. Now I should have the top 25 down regulated genes as well here, and as you can see, all of these have a negative log fold change. Okay, now if you guys remember, I told you guys that we'll be using the normalized counts later. So we will be using the normalized counts here. So before we begin from here, At this point, our differential gene expression analysis is complete. Okay. After this, we will be learning about two very important types of visualizations that we do. Okay. The first is a heat map, and the second is a volcano plot. Okay. So let us first understand the heat map. So in order for us to plot the heat map, first we need the normalized counts values. That is, from the normalized counts, we want only the values of the ones which are part of our top 25 upregulated genes. Okay. In our heat map, we only want to see the values for only the top 25 upregulated genes. Okay. So if I run this, basically what it will do is it will subset the normalized counts df. The normalized counts df, if you guys remember, it is this, right? But it has all the genes. It has all the 13,000 genes, right? I'll close these so that it's a bit easier for us to navigate. Okay. Yeah. So now I want only those genes from normalized counts df which are also in top 25 up LAA control, because I want the counts values, the normalized counts values of only the top 25 upregulated genes. Okay. Now, if I do that, I should have a counts matrix with only the top 25 upregulated genes and their respective counts values, normalized counts values. Similarly, we will also get the normalized counts values for the top 25 downregulated gene. The logic is just the same. Now we will be using the library pHeatmap. So go ahead and load it, right? And basically when you give the data to pHeatmap, it expects it as a matrix. So you have to convert the data frame to a matrix. For that, we use the function as.matrix. Once we convert it to a matrix, then we define the function pHeatmap to basically show our heat map. So in the heat map, basically what will happen is we will see the genes as well as the samples and how well they group together, right? This is basically kind of like a diagnostic test for our analysis, right? It will basically show us whether the analysis we performed is actually fruitful or not. Okay. Let me first run this and then show you guys what each of these means. If I run this, you should get a beautiful heat map that looks something like this. Okay, let it load a bit so that you guys can see. It's not loading properly. Okay, not a problem. You can just use the zoom and can see. Okay, this is how your heat map looks. So what is there basically? First, let us understand how to interpret it. Okay, first thing that I want to actually look at is this legend here. So this basically denotes an lower expression or down regulation, and red is up regulation, and you see a very clear gradient here. So as we go from blue to white and then red, we are going from down regulation to up regulation, right? And this is a heat map of the top 25 upregulated genes. And here upregulated means upregulated in large artery atherosclerosis as compared to controls. As you can see, for the LAA samples which are labeled here, the genes are mostly upregulated. There are some blotches of downregulation here, and that is exactly why in the PCA, if you guys remember, we did not see very clear clusters, and this shows here why we did not see those very clear clusters. But in case of healthy controls, we can see most of these genes are actually downregulated. So a very important inference from this. Okay, whatever will be upregulated in a particular condition, say LAA, should ideally be downregulated in the healthy control, and that is why it is upregulated, right? If something is +2 in the other one, it has to be -2 in the other condition. That is how math works. That is how
Totally.
logic works.
But
Yes.
the thing is,
Mm.
you are showing that upregulated genes, and you are telling that
Mhm.
the +2 will be the upregulated, and the control ones
Yes.
are
Yes. So
the condition one
whatever
will
is
be
whatever
showing
is
downregulated. But
yes.
the thing is, The control one should be the zeros, right?
No, so control. So what this means is these guys, these genes are upregulated with respect to the control. If something is upregulated with respect to the control has to be downregulated with respect to that, right? That is all it shows here. If you were to
No, no,
plot the
sorry. The
absolute,
control will be the baseline, right?
yes, the control is the baseline, correct.
Yeah. So
Now,
whatever you
if
see in the condition in the
hmm
LAA, that's
hmm
upregulated with respect to the condition in the in
condition
the
control,
control, right?
yes, exactly, correct. Let me explain why I'm saying that these would be down regulated per se, or why are you actually seeing minus values for these. Okay, if you were to plot the absolute values of the normalized counts, you would probably not see that. Okay, then you cannot really say that whatever is upregulated here would be downregulated here. But when we plot a heat map, okay, what we do is we do a z-score normalization. This particular parameter, scale equal to rho, does a z-score normalization on the heat map data, that is for the counts. So for each gene, if you guys know what a z-score normalization is, a z-score normalization mean centers your data. So the count for each gene now has mean 0 and standard deviation 1. And this basically helps us visualize the gradient, right? If I were to use absolute values, I wouldn't be able to put a symmetric gradient like this from -2 to 2. Okay, now that I have scaled it by row, as in I have done a z-score normalization and mean centered my data, I am able to visualize the gradient, and I am actually able to figure out that, okay, whatever is upregulated in LAA is actually showing downregulation in healthy controls. And if you think about it, if something has to be upregulated with respect to a control, the control has to be downregulated with respect to that something, right? From where did the concept of up and down regulation came? The up and down regulation concept came from the fact that there were more reads mapped to that gene in the condition than to the control. So if I am saying that there are more reads mapped to condition with respect to control, I can also say there are less reads mapped to control with respect to condition. It's the same thing as saying like that. Your baseline does not mean it is zero. Your baseline does not equate to zero. Your baseline is also some form of expression. But you have set it to zero and you are saying with respect to this baseline, these are upregulated. Does this make sense?
Okay. So I think the chart should be uh, total gene regulation. Then we
Yeah.
expect, if we expect the 25 upregulated and downregulated in a heat map,
hmm hmm
then it should be
So you're
we can understand
so
it better.
you're saying that the heat map should contain both the top 25
Both.
up as well as the top 25 down regulated,
Yeah.
right? Yes, a lot of people do that as well, but in that case, what happens is basically these writings are not as clear. Okay, so
Okay.
people just end up doing top 10 and top 10, total
Okay.
20. Top 10 upregulated, top 10 downregulated. You will see a lot of papers do that. A lot of papers, just for the sake of clarity and just because they want to report a higher number of genes, they also do it separately. And why both of them make sense is because ideally, if you have done your analysis correctly and your conditions do have biological difference, you should see you should see the classes clustered together in both the cases. As I said, when I started explaining heat maps, this is kind of a diagnostic test of how well your analysis worked. Okay, and that is why this is what I personally do. I personally plot them separate because ideally they should separate out properly in case of both upregulated as well as downregulated separately. Okay, it is absolutely fine if you plot them together. It is fine, a lot of papers do that. You will see a lot of papers doing that. It's not wrong. This is one approach. And this also makes sense. It's not like it does not make sense. Okay.
Okay. And tell me another thing.
Mm-hmm.
I just haven't that idea that
Mm-hmm.
here they are showing a thing like
Mm-hmm.
what we see in the phylogenetic tree-like
Yes.
structure.
Dendrogram.
Yeah. So,
Dendrogram. Yes.
is it mean that LA2 and 3 are values more close to each other, and
Yes.
LA1 is the
Exactly.
outlier?
This is known as hierarchical clustering.
Yeah.
Okay, according to the calculations done internally by P heat map, since I have put cluster rows equal to true, this T is basically true, and cluster calls is equal to true, and this is the default. Basically, I have only just showed it to you guys. This performs a hierarchical clustering on your data. So hierarchical clustering is nothing but a form of clustering your data in the sense that it tries to bring together closer data points together and keeps farther away data points farther. It's just what clustering means. It tries to bring together the close ones together. So here ideally, since you are seeing that LA2 and LA3 have the same common branch, right, but LA1 has a different common branch, this you could interpret it like that LA1 is actually a bit more different than LA2 and LA3. You can. But again, this particular clustering technique, it uses Euclidean distances. Okay, so you cannot exactly correlate it with the PCA.
Okay.
Okay, because your PCA does not use Euclidean distances. Your PCA builds those principal components to basically showcase the distances to you. But these guys, they use Euclidean distances. You can change the distance metric as well, which I will come to when we plot for the data down. Okay, for the down regulator. Okay, yeah. And so the rest of these parameters are just for with respect to the with respect to the graphics basically. So show row names true and show call names true basically just shows the gene names and the sample names. This is especially comes in handy when you're actually trying to visualize. Okay. You cannot really make out a lot from this clustering. I mean, you cannot say that SERF1B and the other one. So, for example, these two, for example, PSMB8 and POU3F1 are actually very similar genes. No, you can't really say that because these are just the gene counts, right? These are clustered based on the gene counts. They are not clustered based on how similar or how well these genes are tied together in a biological process. Just because they happen to have high counts together in a sample and low counts together in another sample does not mean that it has a biological significance. It is, I mean, it could mean, but it does not necessarily mean. So don't read too much into the gene level clustering, but this level clustering you could use as part of your interpretation. Similarly, if we were to plot it now for the down regulated genes, uh, we can go ahead and plot it here. The only thing that I want you guys to show here that I have added in a new parameter called clustering method. Okay, so in case of hierarchical clustering, it uses something called linkage, and that is basically what the clustering method is. The default type of linkage is actually complete linkage. Okay, and in case of hierarchical clustering using complete linkage, what we do is we calculate the distance between two clusters as the max distance between any pair of data points, right? So for example, if for example LA3 was farthest away from HC1, so HC1, HC2 and HC3 should ideally form a cluster, and LA1, LA2, LA3 should ideally form a cluster, right? Now to find the distance between the two clusters in complete linkage, we will find the maximum distance between any two data points, right? Say for example HC1 is farthest away from LA3. This is the maximum distance. This could be the maximum distance between the two clusters. Okay. This just basically means that HC1 is the one which is farthest away from the farthest away data point of LAA, which could be LAA3, right? So if you think about it here, complete linkage tries to maximize between cluster distance, and that is why it is not as robust and is actually very prone to outliers. And in our PCA we did see outliers, right? That is why here while plotting the heat map I have changed the clustering method to ward.d2. This uses something called Ward's distance as the linkage method. And Ward's distance basically focuses more on minimizing total within cluster variance. So complete linkage maximizes between cluster variants. Watts distance minimizes within cluster variants. So Watts distance tries to minimize the distance between each sample within a cluster. And that is why it is less prone to outliers. Because if you are focusing only on one cluster at a time, you will be making sure that a single outlier is not actually affecting the distance, right? Right now, if HC1 would be very much farther away from HC2 and 3, it would affect the distance measure if you were using complete linkage. But here, if I use this form of linkage, we are actually minimizing within cluster distance to find the clusters, the appropriate clusters. Um, Again, all I want you guys to take away from here is there are different ways you can perform hierarchical clustering. You can either maximize between cluster distances or minimize within cluster distances, right? If you are minimizing within cluster distances, you're more robust to outliers. Outliers are less likely to affect your clustering. And since we did see outliers, I have used ward.d2 here as a clustering method. Okay, I will now show you the one for down-regulated one. So I have to first run this line where I convert it to matrix. Okay, wait a second. Yeah, where I convert it to a matrix, and then I will perform the clustering. Similarly, here you can see that in the healthy controls you can see kind of plus values, but in the conditions you are seeing negative values, which show that the down regulation is on the condition side, which is what we expect. Okay, now if I were to use complete linkage here, as I explained, instead of ward, you can see that one of the healthy controls is actually coming closer and closer to the LA ones. But when I actually see the heat map, I can actually see that these guys, they are actually upregulated, but in the LA they are not, right? So ideally this should cluster with HC1 and HC3. So what happened was this HC2 might have might have basically pushed itself away when we are using complete linkage, because we are now trying to we are trying to maximize between cluster distances. So your clustering method matters, what clustering method you are using. Okay, the default is complete linkage and works for most cases. For some cases it might not work, right? Then you should use something which is more robust to outliers, which is in our case, which is ward.d2. There is also a ward.d, but the d2 is a like a better version of it, so people utilize that these days. So this is on heat map. Okay, now we have pointed out that the top differentially expressed genes, so the top 25 top 25 upregulated genes as well as the top 25 downregulated genes are able to differentiate between my two conditions. Even if barely, it is still able to do it. If my data would have been much better, I would have seen much better clustering here. You would, I mean, in the most ideal scenario, you would see a completely blue block here and a completely red block here. Okay, that is the most ideal scenario. But again, these are biological experiments data. They cannot be perfect, they cannot be ideal. So it is all based upon your interpretation. Okay, this to me is a good enough heat map. Okay, you can see clear distinction that at least
So
this is
I
not
have a doubt.
yes,
Sorry to interrupt.
yes, fine.
So what is the significance of going with the heat map with normalized counts?
Yes, so if you use, first of all, though, you cannot use the absolute counts, right? Because if you're using absolute counts, you're again you're defeating the entire purpose of normalization, right? What was the purpose
Okay.
of normalization? That you should be able to compare between conditions, compare between samples. That was the purpose of normalization, correct? That is why
Yes.
you have to go with normalized counts. Now you don't even plot the actual normalized counts here. You do another round of normalization known as z score normalization. This basically mean centers your data. What that means is, now every gene's normalized count has a mean 0 across all the samples and a standard deviation of 1 across all the samples. This basically helps us visualize it in a clean symmetric gradient. That is exactly why
Okay.
you do the z-score normalization. So the first
Okay. So
normalization is to make
yeah.
sure you can compare among samples, and the second normalization is to make sure that your visualization is symmetric, so that it makes it easier for you to draw conclusions.
Okay. So both.
Okay.
So it would be same as the DeepSeek result, or would that be any difference?
No, so DSeq is going to give you the normalized counts. So this is what comes out of DSeq. Okay.
Okay.
Z-score
This is one of the
normalization.
results. Okay.
Yes, this is the first layer of normalization. This is what comes out of DSeq. When you use the parameter scale equal to rho, you perform the z-score normalization as well, and that you do inside p heat map.
Okay. So if
Yeah,
we want to report means what type of heat map we need to report, whether
this
this
is the
heat
heat
map
map you
or
need to report. You need
oh,
to
this is
report
the
it
heat.
after z-score normalization, because
Okay.
that is what I have seen in most papers. They always perform the z-score normalization, because so that the visualization becomes more, what can I say, intuitive. Okay.
Okay.
Now
Okay.
directly if I see here, I can check that, okay, my blue means negative, so blue means down regulation. That is very intuitive
o-
for me. Otherwise
Okay.
it is not very intuitive if you use the absolute normalized counts. Try it, no,
Oh,
once. When you try running this, try it without the scale equal to rho, and see if the graph is as intuitive as with scale equal to rho.
yes,
Try
I'll
and
try.
compare.
Yes.
Yeah,
Yeah.
try and
Sir,
compare.
yeah, I have one more doubt. And in
Yes.
papers, I have seen like this is a shrinkage result and this is unshrunken result after
Yes.
these. Yeah.
Yes.
So I tried with the shrinkage and the unshrunken one, but
Mm-hmm.
during shrinkage, I lost so many significant genes, even though
Mm-hmm.
it passed the adjusted
Filter.
p-value and the log fold
Yeah.
change category.
Yeah.
So what should I do? Whether I should go with the shrinkage because even though it passes the adjusted p-value category and also a
Mm-hmm.
log fold change category, that gene
Mm-hmm.
is not coming under when I perform the shrinkage.
Mm-hmm. So again, LFC shrinkage is something which is specific to DSeq2, and this is specific to DSeq2's method. Okay.
Okay.
So since DSeq2 fits a linear model, right, and some genes, if you remember, DSeq2 is very it is very lenient with outliers. It does not remove outliers as such
Okay,
when
sir.
you're doing the normalization and all. If you remember, in none of the steps we were removing any outliers, we were just accounting for them. Okay.
Yes.
But how much accounting is too much accounting? You can never account for all the effects those outliers will cause, and that is exactly why if you have a lot of outlier genes, not samples, outlier genes, you will have log fold change values which are extremely high, so such as 15, 16, 17, 20, 25. You could have very high log fold change values, and that is why DSeq2 has a method to shrink those values down. It performs some more complicated statistical tests. Even I don't remember exactly what it does to do the shrinkage, but it tries to shrink down the log fold change values, okay, so that they are in a respectable range. Okay.
Okay, sir.
In my opinion, and this is what I had done for my latest project, with one of the data sets, I got extremely high log4 change values, and that was very that was very expected, because I had a lot of outlier genes as well, right?
Okay.
hr hr is less sensitive to these things. So what I did was I used hr. I did not use shrinkage, because shrinkage I am risking losing out on information, which is what you did,
Okay.
right? You lost out on
Yeah, yes.
information,
Yes,
right?
yes.
So in that case, I'm not saying this is something that people should do. This is what I did, and it made sense to me, and it will actually make sense to you as well when we do when we go through the theory of how HR normalizes stuff, right? It actually does not account for the outliers. It actually removes the outliers so that your analysis is more outlier safe. Okay, and that is what HR does. That is why HR is different from DC. Okay, so
Oh,
my
so instead of
advice,
DC2, I
yes,
can
try
proceed with
HR.
HR.
Try with
Okay.
HR.
So,
Again,
yes, sir.
if this is a personal project, take my advice, take it up. Okay, if it is a project
Okay.
that you're doing under some professor, or you have a supervisor for the project, please consult with your
Okay.
supervisor.
Sure.
Okay. Yeah,
Sure.
I mean, don't just take
So,
my word for it. If you
yeah,
have a
yeah, yes.
supervisor, consult your supervisor as well.
Yeah, sure.
Okay.
And is it mandatory to report the shrinkage one, or can I proceed or what is is there any rule like we have to go with the shrinkage one, or can I proceed with unshrunken DEGs also?
There is not a rule per se, but think about it. If you have a log fold change of say 20, what does that
Yes.
mean? That means that you have a fold change of 2 to the power 20.
Yeah.
That is a
Yes.
huge number, right? So do you
Yes.
think such a huge fold change actually makes sense? No, right?
Okay.
That is
Okay.
why people go with shrinkage. People are of the opinion that such huge fold changes should not happen in a biological system that is so tightly regulated, right?
Okay. Yes, sir.
You
Yes,
will
I
have
got it. Yeah.
differences, but you should not have as high differences. That is the reason why
Yes.
people
Yes.
go with shrinkage. And in that case, if you are doing
Okay.
with DSeq2 and if you're doing shrinkage, you should report both. And you should also report that you lost this many
Okay.
genes to the shrinkage. And these are the ones that were
Oh,
left.
okay. Fine. Okay. Fine. Okay,
Yeah,
sir. Yeah. Thank you. Thank you so much.
you're welcome. No problem. Okay. The second visualization that we have after RNA-seq, again, this is a very common one, that is a volcano plot. Okay. I will first plot the volcano plot and then I will tell you guys what we did and how we did. Okay. Let me run these 3 lines. Yes. Now it should come. Yes. Okay. So this is what a typical volcano plot looks like. Okay. On the x-axis, you have the log2 fold change. You plot the log2 fold change. On the y-axis, you have something called negative log p-value. Okay. You have minus log base 10 p-value. Okay. This is nothing but your p-adjusted value or p-value, whatever you had filtered with. On that, you basically just perform the function minus log 10, right? Why do you do that? You do that because usually your p-value will be somewhere between 0 and 1, right? That is how p-values are. It will be always between 0 and 1. If you were to plot that, they would be too squeezed, right? If you think 0 and 1, so everything will be squeezed in between this, just in between this cell. So it becomes difficult for you to visualize. That is why you take a negative log of p-value. For example, if I just were to give you an example, a p-value of 0.05 would translate to a negative log of p-value as 1.3. On the other hand, a p-value of 0.001, if you do the math, would translate to a negative log p-value of 3. Now you see, between 0.05 and 0.001, the difference is so low. It will be very close to each other. You won't even be able to see the difference. But a difference between 1.3 and 3 is pretty large. It is a difference of 1.7. The graph is a bit more spread out. It helps you visualize it in a better way. That is exactly why we transform the y-axis. And we don't just plot the original p-value. Okay. Now, what can I make out of this? Okay. So what I can understand from this is that my log2 fold change on the x-axis, I have a negative side as well as a positive side. So genes to the right of 0 are my upregulated genes. Genes to the left of 0 are my downregulated genes, right? Now you can also see 3 colors here, red, blue and black, right? So genes which are basically in black means that they are not significant. They did not pass my p-value cutoff. My p-value cutoff was 0.05, right? That would roughly estimate about 1.3 in the minus log 10 p-value scale. So anything that has a minus log 10 p-value of less than 1.3 is automatically denoted as black, which means not significant. So these are not statistically significant. So only genes which are statistically significant are colored with anything other than black. The blue ones are to the right of 0, so they are colored with blue and are annotated as upregulated. The red ones are to the left of 0 and therefore they are downregulated and annotated as such. The key interpretation from a volcano plot is that genes which are to the far left or the far right, such as genes like this, genes like these, they are the ones which are the most biologically interesting because they showed the highest log fold change, right? genes near the middle are not of the most biological interest, but still, since we put our cutoffs, since we decided on the cutoffs before the example, these are also of interest to us, and we will be using the entirety of them as our for our pathway enrichment or functional enrichment. Okay. Now this is where this is up to what you guys have in the DESeq2 script that I have provided you. Now we can also label the top 25 genes here. Yes.
And the genes that are at the top,
Mm-hmm.
what does that mean?
So these are the most statistically significant. So genes which are particularly at the top had the least p-value. That means, right, on the negative log 10 p-value, the higher the p-value is, the higher your negative log 10 p-value, the more
That
significant it was.
that means that gene show the less false positive, right?
Not I mean, yes, in terms of you understanding, yes. That means that if you were to do 100 experiments
That
with
means
this
100
gene,
experiment, the only
only
the
1% chance is there that this was wrongly called as positive.
okay.
Yeah. Okay. Again, so there is one more enhancement that you guys can do on this. Okay, before I do that, I'll just quickly explain how this works. So here you basically just give it the color scheme that you want to use. Okay, and the color scheme will be based on whatever you give here. And actually before that, we are actually creating these two columns in our SDF, which is the negative log p-value. We are only doing minus log 10 of the actual p-value, and we are also adding a column for expression, right, based on the condition that if it is greater than 0.5 or 0, whatever, you can do that. I have put a cutoff of 0.5 here. If I would change the cutoff to 1.5, I can do that as well. If I change the cutoff to 1, I can do that as well. Whatever log fold change cutoff you report in your document, you should always use that log fold change cutoff in the plot as well. Okay, that is really important. So here we are basically just telling we are basically just creating a column. If you see here in resdf, we will now have a column which has expression as well, whether it is upregulated or downregulated or not significant based on our cutoffs, right? And that is all we are trying to plot here. And this v line and h line are basically vertical and horizontal lines. These are just lines that basically show beyond which you are defining your statistical significance. Okay. Now the other thing that I wanted to you guys to show is so this is not part of your DESeq2 script. This I did before the lecture. You can actually, and I've seen a lot of papers do this as well, and this actually comes in a lot of very handy as well. Now I know that genes like this guy, for example, or this guy, these genes are probably very highly influential towards my analysis, right? So now I need a way to know what this gene is actually. So we can also label them. Okay. In order to label them, all we have to do is we do the plot as it is. So this part remains same, but we also use a function called geom text repel, and the data we give to it are our actually top 25 gene matrix that we created, right? And we basically, since we are using plus between each of these functions, we are basically tying them all together. Okay. Now if I run this entire thing, and then okay, HCLA res not found. Why did we not save HCLA res? Where is HCLA res? Just give me a second, guys. Acha, it should be resdf, not HCLA res. Sorry. Here it should be resdf. Okay. Now if I acha, it's just the variable names which are different here. Let me fix that. It should be 25 LA control. So actually I was also developing the HR script alongside this, That is why the variable names got mixed up. Should be dot 25 down LA control. Yeah. Now it should work. Yes. Now if I plot it, okay, log FC not found again. So okay, there are a lot of variable errors here. Don't worry, I will fix them. Okay, it should be, I think, log 2 fold change. Yeah, variable errors. I'll quickly show you guys in the HR script. Actually, we will quickly run this and I can show you guys the labeled ones, so that just for the sake of showing. Okay, don't worry about all the plots that are coming up, we will be going through them anyway. Yes, this is what I wanted to show you guys. So for HR, for DSeq2 also, you should be getting this. Okay, now I have labeled the top 25 genes on my volcano plot as well, and now I can actually know what exactly these are. Okay, so this labeling, again, this labeling part I will fix in DSeq2, and I will only share the fixed version with you. Don't worry about that, it's just variable names that I have to change. But yeah, the whole point of labeling them is to understand what each of these genes, or the ones which are of most interest to us, what they could mean. Now you could ask that why are these not labeled? Because these did not pass my top 25 uh, top 25 log fold change cutoff. Okay, maybe the top 25 log fold change, so this is somewhere around, somewhere less than 2.5. Maybe my top 25 down regulated genes, none of the values are greater than -3 maybe. That is why these did not get annotated. You can annotate them as well, don't worry about that, you can annotate them as well. Okay, maybe I can provide you the code to do that as well, because these are also stuff which are actually very interesting. So you can annotate both based on the top 25 as well as based on your p-value, the ones which are the most significant. Okay, so this is how we perform differential gene expression analysis from DSeq2. Finally, you will be left with a table or a file that should be there in your data directory that
Surajit,
has all the
what
results.
file you have shared with us
Mhm.
doesn't have this thing.
Yes, it does not have the labeling part yet. I will fix it on the DSeq2 script. As you saw, some of
Okay,
the
okay.
variable names were not working. Yeah,
Yeah.
I will fix it and I will share the latest one with you guys after the lecture for DSeq2. Okay.
Surajit, another my query is
Mm-hmm.
in the just
Yeah,
in the line number 250, I think.
250 for the previous script,
No, no, this one.
DSeq2 script.
Yeah, this.
No, but 250 is not there. This is where it ends.
Okay.
I think you were talking about the previous script only. Wait, because I was on that
DC2.
a while ago. DSeq2 only.
Any, yeah, DC2.
Acha, you would
120,
you
120, line number 120, or
120, yes.
where you actually plotting this volcano plot.
Yeah, this is the part where I'm plot. This is where I'm defining the volcano plot. Okay,
Uh,
using the ggplot syntax, and this is where I'm plotting it in 130.
yeah, the
Hmm.
before the volcano plot
Mm-hmm.
equal to GG plot,
Mm-hmm.
you just define the data frame.
Yes.
Yeah.
So.
So in this data frame,
Mm-hmm.
these red dots or blue dots,
Mm-hmm.
uh, you just define that race underscore df p
Mm-hmm.
value is less than equal to 0.05.
Yes.
So is it less than equal to this will be the minus log 10, isn't it?
No, no, no. So my
Okay,
definition of
okay. You just first. do that screening. After that, you push that
Yes.
p
So the
value to log.
whole point of converting this to minus log 10 p-value is so that the data is spread out enough for us to visualize. It has nothing to do with the scrutinization part.
Okay.
Okay. The scrutiny will always be based on p-value,
Okay.
but
Yeah,
so
yeah.
that we can
After you
visualize.
take that thing and
Yes.
convert that thing.
Yes,
Okay, okay.
exactly. Yeah, yeah, yeah. Okay. I don't think we'll have enough time to cover HR today. I know I missed yesterday's lecture, so my bad on that. Okay. Does anybody have any more questions with regards to DSeq2? I don't expect you guys to understand every line of the code right now. I know a lot of you know R. For them, it might be easier. A lot of you have no idea what coding is. That was me as well 2 years back. But now I understand almost all of it, at least what I have showing you here. So don't worry. On the first day, you won't understand every line of code. You will get there. Slowly but surely, you will get there. Don't worry. But as long as you understand the idea in the analysis, I am good.
So another question is
Yes. Yes.
when you do that, uh, when you just uh, discard those elements, I think the observation came to a number of 19,270.
Those were the total number of genes we started with.
Okay.
Then we discarded the ones which did not follow our
Yeah,
filtering
the
criteria.
normalization count, DF, data frame. Okay.
Not
After that, you make that 25 observations. Okay.
ha, the 25, the top ones, and
Top
that
one,
is based on my log fold change.
log fold change.
Yes, as long as it is statistically significant. So it also has to pass the p-value less than or equal to 0.05 criteria.
Okay.
Yeah, but we don't order it based on the p-value, right? If you look at it, we have ordered it, we have arranged it based on the log2 fold change. Okay.
Okay.
It is also, you can also order it based on the p-value and keep only the ones which are the topmost ones, the topmost statistically significant ones. That is also a very unique approach that a lot of people do. They take, right? So ideally, if you were still in the experimental stage, you could do the arrangement based on both log2 fold change as well as p-value, right? You could try both and see if the top 25 have any intersections between them. So those ones would actually be your true significant ones if you look at it from the point of statistics, right?
Okay.
Yeah, people do n number of things to experiment and see how they can get some results. So everything is allowed as long as you say that you have done that at the end of the day. Okay.
Another thing is when you do that heat map,
Mm-hmm.
you just added an extra line clustering method for the downstream,
Yes,
because
down
the
regulating.
without that line, the clustering, the data
Mm-hmm.
is it's not right, somewhat different.
I wouldn't say not right. uh, it's not clustering as per my liking, right?
Yes, yes.
Ideally, cluster healthy controls together and LAA together. So it
Yeah,
did
yeah,
not
yeah.
according to my liking.
Yeah. So if I face that kind of problem,
Mm-hmm.
so then I used this word dot
D2.
D2.
Mm-hmm.
Okay.
It is absolutely fine. You can
Okay.
use that, but you should report that since we were facing this issue, and in the PCA we actually saw that there were outliers in the data. So we
Okay.
went ahead with word.d2 because that is less sensitive to these outliers.
Okay.
Yes.
So is there any other clustering method?
Yes, there are several clustering methods. There is
Okay.
something called centroid. There is something called single. So all of these have its own perks, have their own pros and cons. Okay.
Okay.
You can actually, so basically what pheatmap does is pheatmap uses the hclust function of R. Okay, it is just pheatmap is a wrapper around hclust. So you can go into the H-cluster documentation. I can actually show you. I have it open, I think. This is P heat map. There is also H-cluster. You can go into the H-cluster documentation and check that out.
So I have
Okay.
seen that heat map 3. So P heat map is the latest one, or
So P heat map, what do you exactly mean by latest one? I
I
did not
mean to
understand.
say have more features, or have there are any problems that is solved and included in this one?
There are several ways to draw heat maps. P heat map is the most common one that I have seen being used for RNA-seq data. That is why, I mean,
Okay,
even I
okay.
use P heat map, and you have so much control, right? You have control over whether you want to cluster the rows, whether you want to cluster the columns. You can put any kind of so this is with respect to the clustering method. Okay, there are other parameters
I
here
think
as well.
I used heat map 3.
Heat map 3. Okay,
Yeah.
that also works. As
Okay.
long as you have control over the methods you are using, right? Your function or your tool should give you the flexibility to bend it according to your will. Okay, that is all we do in data science. We bend the data according to our will. So you should have that flexibility. As long as your tool gives that, you can go ahead with that. Not a problem. Do you have in heatmap 3? I have never used heatmap 3, so I don't know. But do you have control over what type of clustering mechanism you want to use?
No, I didn't see that thing clustering methods.
Okay,
Uh,
use the defaults.
I just hear first time from.
Okay, okay, not a problem. Heatmap 3, if it is an R, it's an R package, right?
Yeah.
Yeah, it should also have parameters for you to choose what clustering method you want. You can check out into the documentation. Okay, it should have the parameters. Ideally, it should. If it does not, you can always use pHeatmap.
Okay.
Okay, yeah.
But actually, about the level you change in the gene expression analysis, the
About
name
the
of the genes. Yeah, name of the genes. How do
name of the
you
genes, yes.
aid that? Yeah.
How
So
to?
can you like how do you aid the gene name of the genes? Like
How
when
did I
I
label
run,
it here?
yeah, yeah, yeah.
Yeah. Okay. So this is what I was explaining then. You define the volcano plot as is, as you defined earlier. Then you use this function called geom text repel. Okay. The first thing that you give to geom text repel is basically you give it the data, which is your top 25 up LA control. This is the one that has the information on which are your top 25 upregulated genes, right? Then you define the X and the Y axis. So this is basically, think about it as when you run this, when this part of the code runs, right? This, if I can show you one of the earlier volcano plots, can I cannot. Okay. Just imagine that once this part of the code runs till here without the plus, this volcano plot is rendered but without the labels. Okay, now when you run this part of the code, you put the labels on top of this volcano plot for the top 25 upregulated. If you see here, my AES definition is the same for both, right? X is log FC and Y is negative log P value or whatever, right? And my label, I have just changed it to top 25 up HC LA. So you are basically, first step is you render the plot first without the labels. Imagine none of the labels are here. Second step is you add the labels for this guy. And third step is you add the labels for the downregulated ones. Same logic here. Okay, imagine it like that. I know the syntax looks very difficult. It is very difficult to me as well. If you tell me to write this without looking at it, I cannot at this point.
Sir, over
I will
here,
be very honest.
what does the max dot overlaps function does?
Yeah, so the max dot overlaps function basically allows only 25 overlaps, in the sense if two genes are very close together, right? And the actual names overlap. For example, twist2 and TUBB2B, they might overlap, right? because they're very close. So it allows for 25 such overlaps. Okay, beyond which you won't be able to see. Even as a matter of fact, you can see that when you plot it, you get a warning message as well. The 13 unlabeled data points, right? That means out of the ones that I wanted to label, 13 of them have been left unlabeled. The heat map has just not labeled it. Why? Because there were too many overlaps, and if I have too many overlaps, I won't be able to read, right? If this overlapped with this, how will I read? That is what this means. That up to 25, there should not be any overlaps. Okay.
Okay, sir. Thank you.
Yeah, but still, if R feels there will be overlap based on its graphics device, it will tell you that there will be overlap. So I won't label these 13. It will simply tell you. If you want to bypass it, you have to increase that value. Yes, so Dr. Pinake, I think you were trying to say something.
I just want to know that this 25 haven't any relations to the 20. We have 25 representatives, so we use 25. That is not the thing, right?
No, you can change it as well. It's not like you have to use the number 25 only. You can
No.
use.
That not that max
Okay.
overlap equal to 25.
Oh, no. This has nothing to do with that we have 25 genes. This is just I think the default only is 25.
Okay.
That is why I put 25 here. Okay. This was, I mean, I took all of these parameters I took from the documentation. So the default only is 25. Yeah. Okay. I think we are already quite a bit over time. So let us have a quick discussion on how we are going to move forward. So Bhavni, I guess you are supposed to take a poll, right?
I just announced in chat that tomorrow's session will extend a bit, say 30 minutes.
Huh.
Uh,
Okay.
Shall we take one tomorrow for next session?
Okay,
I mean,
sure.
if needed, if participants are requesting for us to take a session, maybe that can be asked in tomorrow's poll. But anyhow, tomorrow's session will extend 30 minutes because we need to announce a mini project, right?
Yes. So guys, tomorrow I will be announcing the mini project. So we will spend some time on that. But in that case, so I just want to discuss this with them once, because I feel like, I mean, it's just my way of trying to make amends for yesterday. So you guys saw how much time it took for DEC2 today, right? Tomorrow we will be doing HR. Okay. If we have to maintain the flow. If we do HR tomorrow, we will, if I do the theory as well as the practical, it will take up the entire 1.5 hours. A lot of the stuff is same in HR, such as the volcano plot, heat maps, everything is same, so I won't re-explain them. I will only explain the HR part, right? So that won't take much time, but the theory will take again some time. So HR will take some time, maybe 1 hour, the entire HR thing. Okay. After that, we can have the mini project discussion, which will be another half an hour. So we can end it at 8:30. And then we will be left with a topic called as functional enrichment, right? For functional enrichment, I will need maybe another 45 minutes to an hour. So that won't be possible tomorrow. Correct, right? So, I mean, the idea was that initially what I had thought is I would take another lecture on Sunday, right? But a lot of you might not be available Sunday. So what do we do now? That is the question. Bhavni, so you were supposed to take the poll whether they want a session on Sunday, right? That was the poll that,
Yes.
yes.
Yeah. But anyhow, tomorrow's session will extend a bit, right?
Yes, anyhow, tomorrow's session will extend
Okay.
a bit. Another request, if it is possible, we can extend it half an hour from 7:30 to from 8:30 to 9, and
Okay.
if we can also prepone it a bit from maybe 6:30.
Suryadeep,
We
I
will
don't
give
think so. That will be a good idea. Why? Because as per schedule, our session is 7 to 8:30. I think
7.
many
I understand.
participants might be planned accordingly. And also
I
tomorrow is a
understand.
Friday, so everyone will be at the work.
I
So
understand.
let's make one
I
thing.
mean,
It's my
uh,
suggestion. If participants also quite interested in doing that, not an issue. Like maybe we
mhm,
can just take tomorrow's session till 8:30, and it may last for another 30 minutes till
mhm,
9. And
mhm,
we should also announce about the mini project
mhm,
because weekend I think participants can work on that.
mhm,
After coming to new water, all the topic will be left behind. You just
mhm,
say us, there's more minutes you need to complete the session. That can
mhm.
be compensated in Sunday session. Okay.
Okay.
I don't think so 7 to 8:30 is needed on Sunday. Only
No,
after
no, no.
meeting what are all the topics left out after tomorrow's
Mhm,
session, we can take a
mhm,
wise call. If it
mhm.
is for a half an hour session or it may be an 1 hour session. Based on the topics left out, we can have
Yeah,
it on Sunday.
yeah,
I
yeah, yeah.
think participants will be convinced with this. Please
See,
give thumbs up if you are okay with it.
Ma'am, you can
please.
Then
also
just
take on
yes.
Saturday.
Saturday actually, Dr. Binik, I am not available.
Okay, okay.
So yeah, that is
I think
again my bad.
we hardly need 30 more minutes, Dr. Pinaki, on
Okay,
Saturday.
okay.
I do
Yeah.
understand it's quite
See,
hard, but
if
unfortunately, yesterday was a bad day for all of us. We don't have any
No,
session.
no,
if
it's
Only to
Sunday if
compensate that
Sunday
very.
is too much of a problem for everybody, we also have a doubt clearing session on Tuesday, right? We can spend 30 minutes. I can explain the functional enrichment part to you on Tuesday, and the rest of it we can spend to clear all your doubts. And
Yes.
bear in
And
mind, these are
usually,
doubts
yes,
with,
Surya,
yes,
usually doubt clarification will hardly takes us 30 minutes.
yes,
But it's since it's on 1.5 hours, which we planned
yeah,
already, maybe you can take that part on
yeah,
Tuesday
that
session.
also
I think
works.
that won't be overloaded for the mini project because
No, no.
Tuesday we have doubt clarification, followed by that we have a mini project in couple of days. Won't it be
Yeah.
overloaded if you explain few things on Tuesday?
No, actually, so why I'm saying this is, uh, Tuesday, if we do the
Hmm,
doubt
hmm,
clearing will be completely with respect to the mini project only, right?
exactly.
Yeah.
Yeah.
So we won't need to spend too much time, because that is the entire point why I'm also taking 30-45 minutes tomorrow for the mini project, so that I can explain to them clearly what I want from them. Okay.
Okay.
Um, and again, the functional enrichment part, everybody, that is something that you'll be doing at the very end of your project. Okay. That is why I am still okay with taking that on Tuesday. If Sunday does not work for everybody, if Sunday works, we can get it done on Sunday, and then Tuesday we will have it as we have the doubt clearing. Okay.
I think so. I think maybe in tomorrow's session, let's have a poll asking participants
Sure,
whether they are
sure,
okay
sure,
with
sure,
Sunday session or on Tuesday. We
sure.
also request you to say how long it will take. Okay, based on that, we can
Yeah.
ask
So
participant suggestions and
if
take
it's
a
only
voice call.
if it's
Yeah,
only Tuesday, okay, let us do one thing.
yeah.
Tomorrow, before we begin, let us have the poll, because I think at that time most of the people are there.
Okay,
Okay.
okay.
Yeah. Before we actually begin, so when there are actually like 30,
Yeah,
40
yeah.
people,
That
let us
that
take the
can be
poll then.
sorted out. Yeah,
Yeah. Okay. Okay.
fine, fine. Yeah. Thank you, Surya.
Otherwise, yeah, yeah. Otherwise, we
I think
can
this
think of it. Not a
decision
problem.
can be taken tomorrow
Tomorrow, yeah,
because now
we'll
we
do that.
already lost few of the participants.
Mm-hmm.
We can just make it by tomorrow. We can get
Yeah, yeah.
suggestions from our friends and
Sure,
we
sure,
can take
sure.
a wise call on that.
Sure, sure,
Anyhow,
sure.
tomorrow's session will last for at least 30 minutes. We need 30 minutes extra. I'm saying at the
Extra.
maximum we need 30 minutes. We will
30
end
minutes.
up even before that
Yeah, yeah,
because towards the end we will be announcing the mini project. So this is the schedule as of now, and we are extremely sorry. This is a unplanned one. I know it's not as per the schedule, but guys, please try to understand. It's totally unplanned due to bad weather in Suryadip side yesterday.
yeah. I mean,
So,
completely my bad, guys. That is, I really apologize. I know it's unacceptable. I had nothing to do in that scenario.
yeah, yeah. Thank
Yeah.
you. It's okay. Not an issue.
Anyway,
It happens to everybody, but still,
yeah.
we have quite a range of participants. Yeah,
Yeah. Okay. So then let's have the poll tomorrow
sure.
before
We
we
can
begin.
have it.
Yeah.
Yes, either
Okay.
before we begin or towards the at I think maybe around 7:30 Indian time. Most of the participants will be active
Yeah.
in that time. Maybe we can have that
Yeah. Yeah.
period.
Yeah.
Yeah.
So then I will go on with the lecture. You on in the background, you conduct
Yeah,
the
yeah.
poll.
That decision can be taken.
Yeah.
Yes. Yeah.
Okay. 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 Thank you so
And
much,
thank
Bhavani.
you, dear participants. Yeah, thank you,
Yeah.
Sugyadip. And dear friends, please to fill out the feedback form. And as mentioned, please be available tomorrow at least for 30 more minutes. We can wind up with the mini project in tomorrow's session. With this, I think we can wrap up for today and meet you all tomorrow. So, Suryadip, do you want anything to add on? I think we
No, no,
was
no.
trying.
I'm good. Yeah, we can wrap up. Okay. Thank you.
Thank you,
Thank you,
guys.
everyone.
Thank you so much.
Yeah.
Thank you all.